This is the model of mid-sagittal section of head and neck, showing pituitary and pineal gland. And now we will learn the pineal gland. This is our pineal gland. It hangs from the posterior wall of the third ventricle and is a part of epithalamus, which is the part of the diencephalon. It is a rice grain sized neuroendocrine gland which has got no blood-brain barrier. There are many structures in the head and in the brain which has got no blood-brain barrier, that are organ vasculosum, median eminence, area prostrema, neurohypophysis, pineal gland, subfornicial organ and subcommissural organ. So pineal gland has no blood-brain barrier. It is of rice grain size and its dimension is 7 into 6 into 3 millimeter. That its length is 7 millimeter, breadth is 6 millimeter and the thickness is 3 millimeter. It is also known as epiphysis cerebri because it lies above in the its superior level at the cerebral hemisphere. Okay, and also known as conarium. The word conarium comes from a Greek word konos. Konos means pine cone like. Its shape is pine cone like. That's why called conarium, and also known as third eye, because the main constituent of this gland, that is the pinealocyte, is supposed to be the modified photoreceptor cells, because it shares the common antigen which the retinal epithelium has got, that is the opsonine, recoverin, retinal S antigen. Okay, so it is a midline structure, the part of the epithalamus, our pineal gland. It is reddish gray in color and it is of rice grain size, piriform shape. It is developed from the dorsal part of the roof plate of prosencephalon. As far as the gross structure is concerned, it is made up of one body, pineal body, and stalk. Stalk are two, that is the ventral lamina and the dorsal lamina. This one is posteriorly placed, so dorsal lamina, and this is somewhat anteriorly, so this is ventral lamina. Dorsal lamina, this one is the dorsal lamina of the pineal gland. It is continuous with the habenular commissure. Habenular commissure connect the two habenular nuclei as well as the interpeduncular nuclei. Where is the posterior commissure, which is continuous with the posterior lamina of the pineal stalk. It contains the commissural fiber of medial longitudinal fasciculus, pretectal nuclei, Edinger-Westphal nuclei, nucleus of dark switch, posterior thalamic nuclei, and as well as superior colliculus. The fibers of both sides crossing each other. Here is the commissural fiber lies, which is called posterior commissure. Now come to the relation. It lies posterior to the third ventricle. So just anterior to the pineal gland there lies the pineal recess of the third ventricle. Above it lies splenium of corpus callosum. This is our corpus callosum. This is rostrum, genu, body. Here is the isthmus and the splenium. So just below the splenium there is pineal gland, And also this blue structure, this is great vein of Galen. And below the pineal gland there is, this is superior colliculus. Superior colliculus, here it is. Superior colliculus, here it is. Superior colliculus, the part of tectum of midbrain. And also below there is subcommissural organ. This is posterior commissure, and just below there is subcommissural organ. It is extended up to the terminal ventricle, the ventricle which lies in the conus medullaris, the terminal part of the spinal cord, the subcommissural organ, and it is committed to maintain the CSF composition, which is necessary for the neuronal survival, and also the water balance of the CSF, which I have not made here, subcommissural organ. Okay, it is also in inferior relation of the pineal gland. Now come to as a histology. If we see by the light microscope, then 95% of the cell of pineal gland is pinealocyte, and only 5% are glial cell, that is the supporting cell. Pinealocyte, here we can see in the histological slide, there are some light stained and some dark stained. The light stained are type 1 pinealocyte and dark stained are type 2 pinealocyte. Type 1 pinealocyte is more in number, whereas the dark stained are less in number. Type 1 pinealocyte, which are lightly stained, it contains lots of mitochondria and lysosome, showing its autophagocytic nature as well as highly proliferative nature, highly active stage. And type 2 pinealocyte contains more centriole and the ribosome. Type 2 pinealocyte forms the melatonin, whereas the type 1 forms serotonin, which finally convert into melatonin. Okay. Now one thing more here is in blue color, this is brain sand or corpora arenacea. This is the calcium phosphate and the magnesium phosphate and carbonate here consolidate here to form the brain sand. It increases with the age. Actually, the pineal gland, it increases in size up to the puberty. Afterwards, it do not increase in size, but its weight increases due to the accumulation of this brain sand, the calcification done over the pineal gland. There are many structures in the brain in which the calcification occur, like pineal gland, falx cerebri, tentorium cerebelli, superior sagittal sinus, then choroid plexus, habenular nuclei. All these are structure which become calcified with age. Okay. Now this calcification, this is called corpora arenacea, and in more than 50-year age group, more than 85%, or you can say 85% of person which is more than 50 year, this shows the corpora arenacea, and it is a very important radiographic finding in X-ray of a midline structure. As we all know that the pineal gland is a midline structure, so the calcification of the pineal gland justify the median position of median structure position in the radiograph, and if there is a shift, it will be very obvious that the calcification shift towards the space occupying lesion. Okay. And one thing more, this is in light microscopy. And if we see by the silver impregnation method, then 5 type of cells found in the pineal gland, that is pin, 3P, 1I, 1N. 3P are pinealocyte, then perivascular phagocytic cell, peptidergic like neuron cell, I for interstitial cell, and N for neuron. Okay. So these are the cells which found in the histological slide after silver impregnation method. And one thing more which is characteristic about the pinealocyte is the ribbon synapse. Now what is ribbon synapse? The ribbon synapse, there is a dense granule in the cytokaryon of pinealocyte. There is a dense granule as well as in the cytoplasmic process, which attracts secretory vesicles, which gather secretory vesicles and made available for the calcium to bind for the rapid neurotransmission. And it is also present in the bipolar retinal cell as well as the photoreceptor cells of the retina, the vestibular end organ, the hair cells of organ of Corti, and last but not the least, pinealocyte. All these structures contain ribbon synapse, which will be evident by electron microscopy only. Okay. Now come to this is all about the histology. Now come to the regulation and secretion. First come to function. What does it do? It maintains the circadian rhythm, maintains the sleep-wake cycle. That with the darkness we sleep, when the light fall on us we wake up. This cycle is maintained by a hormone that is called melatonin, which is secreted by the pineal gland. So it maintains the sleep-wake cycle or circadian rhythm. It is anti-cancerous and antioxidant in nature. It is neuroprotective. It prevents neurodegeneration. It is a connecting link between the physical and the spiritual world. Its secretion gives the dream-like state. The near-death dream-like state is visualized by this gland secretion. And also it is effective in seasonal affective disorder. In winter or in darkness, there will be depression due to the low melatonin level. So it is responsible for the seasonal affective disorder. It is also used for the mood elevation. The serotonin is used for the mood elevation. Okay, and these are the function. And now come to the regulation and secretion, which is done by the two methods, molecular control method and neuronal control. Molecular control, it starts with the tryptophan. Tryptophan, which is an essential amino acid. Essential amino acid means that amino acid which is not formed by our body. It is abundantly present in many diets like chocolate, oat, fish, poultry, red meat, almond, peanut, like this. Tryptophan, after hydroxylation, convert into 5-hydroxyl tryptamine or serotonin. And now this get acetylated in presence of an enzyme called AANAT. I am from the anatomy department, and this enzyme is AANAT, ANAT. Aryl, alkyl and acetyl transferase. It is a rate limiting enzyme. It only activates with the darkness. Okay, and it is performed with the presence of cyclic AMP and calcium, and also with the protein kinase A enzyme. And this cyclic AMP acts upon the alpha 1 and alpha 2 adrenoceptor. Okay, so this acetylation process will result into the 5-hydroxyl, the acetylation of 5-hydroxyl tryptamine, which will convert into the 5-hydroxyl tryptamine is serotonin. Now it gets acetylated and acetyled serotonin. Now the methylation process occurs in presence of HIOMT, that is hydroxyindole O-methyl transferase. Okay, the methylation process occurs through which serotonin will convert into melatonin. So this is the molecular regulation, molecular control. And here is the rate limiting enzyme is AA-NAT, which is only activated with the darkness. So with darkness, melatonin is produced. And the neuronal control, it starts with the, as soon as the light fall on the retina, through the retinohypothalamic tract, this message that the light is falling is conveyed to the hypothalamus, the suprachiasmatic nucleus of the hypothalamus. This falling of light, not only on the skin, not only on the vertebrate retina, but also on the GIT and the skin, it senses through the local receptor, that is the melatonin 1 receptor and melatonin 2 receptor, MT1, MT2 receptor. And this message is carried by the biological clock gene, which is NUR77, ERG3, CFOS, AMPER1, AMPER2, zinc finger peptide. These are all biological clock gene which carry the message from the local receptors to the suprachiasmatic nucleus of the hypothalamus, which is known as biological clock pacemaker. And now from the suprachiasmatic nucleus, this information goes to the paraventricular nucleus, and also from both sides it travels to the goes to the T1 T2 level of a spinal cord, intermediate lateral nucleus of the T1 and T2 level through the hypothalamo-spinal tract. And at T1 and T2 level, that nucleus is called ciliospinal nucleus of Budge. And from there, the sympathetic fiber goes to the superior cervical ganglion. After ascending inferior and the middle cervical ganglion, it reaches the superior cervical ganglion, from where the sympathetic nerve go to the pineal gland through the sympathetic nerve, which is known as nervus coronari. Very important, sympathetic nerve of the pineal gland is nervus coronari. And this pineal gland is lined by sympathetic. This is lined by the pyometer. This is lined by the pyometer, the capsule of the pineal gland, and there is traversing trabecula within the substance of the pineal gland. There lies trabecula, which carries the arteriole, venule, as well as the nervus coronari. Okay. So this is our neuronal control, and this nervus coronari, it activates the alpha 1 and alpha 2 adrenoceptor, which secrete cyclic AMP. And this is responsive also. This also insert level where the NAT works, that is the aryl alkyl and acetyl transferase. Okay, so this is a closed loop like autoregulation. This alpha 1 and alpha 2 adrenoceptor activates with the nervus coronari, and the cyclic AMP releases and the acetylation process occur. Okay, so this is all about the regulation and secretion. Now coming to the blood supply. It is supplied by the pineal branch of the medial posterior choroidal artery. Pineal branch of the medial posterior choroidal artery, which is the branch of the posterior cerebral artery, and which again the branch of the basilar artery. Okay, the nerve supply is by the sympathetic nerve. We all know is the nervus coronari, and the neurotransmitter is the neuropeptide Y and noradrenaline. And the parasympathetic innervation is by the trigeminal ganglion, otic ganglion, and the pterygopalatine ganglion, and also through the central stalk, direct through the CSF. Okay, and here the neurotransmitters are in the trigeminal ganglion. The neurotransmitter are neuropeptide Y, calcitonin gene related peptide CGRP, and the PACAP, that is the pituitary adenylate cyclase activating peptide. And in otic and pterygopalatine, the neurotransmitters are the VIP, that is the vasoactive intestinal peptide, and the PHI, the phenylalanine histidine isoleucine peptide. Okay, and through the central stalk, the neurotransmitters are GABA, orexin, oxytocin, histamine, serotonin, and vasopressin. Okay, so this is all about the innervation. And now what this picture says. Here we can see that this person is sleeping well, and this is a born blind child, and it is awakened at night. So sleep-wake cycle is not working properly, because in born blind there will be no gene expression for the light receiving. The 11th and the 15th day of postnatal life, 11th and 15th day of the postnatal life, the gene responsible for the light receiving is expressed. That is NUD77, ERG3, AMPER2, AMPER1, zinc finger peptide, and CFOS. All these are expressed in the 11th and the 15th day of postnatal life. So in born blind, these genes are not expressed. So sleep-wake cycle is not maintained. And what is seen in this picture, that this person has got children, but these blind people, they have no issue. Okay, because the melatonin, this is a blind person but not born blind. This is the blind person, and here the gene has expressed, but there is no light, and there is more darkness. So there will be more melatonin production, and melatonin has got deleterious effect on the hypothalamogonadal axis. So there will be less reproducing capacity. Now coming to the clinical significance, pineal gland suffering with the tumor is called pineal gland tumor or pinealoma. It will suppress the superior colliculus. It can suppress superior colliculus and as well as the medial longitudinal fasciculus, which will result into the Peri-Nord syndrome. In Peri-Nord syndrome, there will be superior gauge. The superior vertical gauge will be affected, and other movement of the eye will not be affected, because the superior gauge, the fiber responsible for the vertical superior gauge, lies on the medial most part of the temporal fiber from the optic tract, and which is relaying over the medial longitudinal fasciculus. So it will get compressed, resulting into the Peri-Nord syndrome. Okay, and due to the compression over the posterior commissure, this will result into the Argyll-Robertson pupil, that is the accommodation reflex present, Argyle Robertson people, but the accommodation reflex present, but the light reflex will be lost. Okay. Now third thing is that due to the pinealoma, it will give pressure over the mid is the